Okay, so I hope that all of you had a good interaction and time to for further discussion. After the talk on technology and zoonosis by Professor Breeze, we now move on to our next eminent speaker, Professor Ruta Mulherkar, who was a former PI in the genetic engineering laboratory at ACTREC, Tata Memorial Center. Professor Mulherkar joined ACTREC, which was the erstwhile Cancer Research Institute, Tata Memorial Center in the 80s. She has really given a very brief CV, which I would want to elaborate further on. She worked in the field of growth factor modulators. Apart from that, she's been working, worked on genetic susceptibility to head neck cancers, gene therapy, and cancer genomics, especially in the cervical cancer. She has made and been lauded with the first transgenic mouse model in the Indian scenario, and which is being still used as a model for hair follicle stem cells. For this work and various of her work, she has published in very high impact journal, including Nature Genetics, Oncogene, FASEP, FEPS, and so on. She is the editorial board member on human gene therapy and various other journals like Journal of Biosciences, Biotechnology, and chairperson of several committees of ICMR, DBT, and BIRAC. I've been discussing, I was fortunate to have gone to receive her, and she is my mentor for PhD. All her students have been in various fields working in different aspects of cancer biology. She is a mentor par excellence and keeps encouraging her students to pursue various facets of research. Today we'll be talking about gene therapy, which has been one of her very big and pet projects that she worked especially in the oral cancers, and worked especially on the adenoviral vectors and AAV vectors for the suicide gene therapy. So we welcome Madam for her talk, and we hope that we'll be enlightened with whatever she has to say. Thank you.
Slide. I can take the slides myself. कौन सा फॉरवर्ड के लिए निकालने के लिए ये फॉरवर्ड करने का फॉरवर्ड ओके Good morning, or rather good afternoon to all of you. And before I begin, I would like to thank Dr. Goyal and his group for inviting me here and giving me this, I don't know if it's, can you hear me behind? Yeah. So for giving me this honor of being the chief guest at this seminar cum workshop. And I also congratulate the group for organizing such a timely, nice workshop on molecular diagnostics. So I'm going to talk on gene therapy, and what I'll do is I'll give you an update or the present day scenario on gene therapy and talk a little bit about the work that we have done. And by the end of the day, by the end of my talk, I hope I'll be able to convince you that this is an emerging novel modality of treatment. So I don't know how many of you know, but the first gene therapy trial, gene therapy is right now mostly in a experimental stage, and there are very few gene therapy products that are already in the market, and as we go along you will see this. So you must be knowing, you must have heard about the first gene therapy that was approved in the clinical trial that was approved in 1989, and this was for a monogenic disorder, severe combined immunodeficiency. And since then there have been more than 2,000 trials for various indications which are being carried out, and these studies have proved provided the proof of principle, but safe and effective gene therapy for genetic disorders is available only for a few. As I said, it's still in an experimental stage. In addition to monogenic disorders, so initially it was felt that gene therapy would be most amenable for monogenic disorders, where you know there's a single point mutation or a mutation in the gene, and the bad gene can be replaced with the good gene. But there are a lot of clinical trials which are going on for disorders like cardiovascular diseases, cancers, diabetes, and so on. So, oops, I'm going back. Okay, so if you look at this pie chart, this gives us the number of different indications for which gene therapy trials are going on, and we find that cancer has the maximum number of clinical trials going on. Almost 65% of the clinical trials that are going on today all over the world are for cancer, and followed by monogenic disorders, infectious diseases, and so on. So what is necessary for gene therapy? You know, gene therapy is you are trying to introduce a gene as a drug into the patients. So what do you require for this? We require efficient vectors, that means vehicles to carry the gene into the cells. We need the gene, the therapeutic or a corrective gene, and we need stable long-term expression of these genes at the cellular level or at the organ level. And we also should be able to re-administer this vector or gene into the patients without causing any problem to the patient, and there should be no toxicity, of course. So if you look at the different vectors, most people in clinical trials are using viral vectors because they are the most efficient vehicles to transfer the genes. You know, viruses have evolved over so many years, and they are able to use the cellular machinery to their advantage very efficiently. And some of the commonly used viruses which are used as vectors are adenoviruses, DNA viruses, adeno-associated viruses, retroviruses, even lentiviruses which cause HIV. These have been used as vectors. And then there are non-viral vectors like liposomes which can transfer plasmid DNA into the cells. So again, if you look at this pie chart where which gives us the vectors used in gene therapy clinical trials, we see that adenoviruses, retroviruses, and naked plasmid DNA, these form the major chunk of the vectors that are used in gene therapy. And so this is in 2017, but if you looked at the pie chart 20 years ago, it was mostly retroviruses that were used as vectors to transfer genes. So coming back to the first gene therapy trial, which was carried out in 1990, what they did, and these children, both of them did very well. This is at the end of 3 years. You see, these are the two girls who were just 4 or 5 years old when the therapy was started, and these are the pioneers of gene therapy. This is Dr. French Anderson, Dr. Michael Blaze, and Dr. Ken Culver. Dr. Ken Culver, we had the good fortune of having him in CRI, Cancer Research Institute, and he helped us set up our gene therapy lab. And this is after 23 years. This is in very recently in 2013. So these two young girls who were kids, they're really grown up now and they're leading a normal life. So to some extent, we can say that this gene therapy has been successful. Now, at that time, as I said, they were using the T cells, and T cells have a limited lifespan. So to overcome this problem, Alan Fisher and his group, they used the hematopoietic stem cells, which are self-renewal, which have a self-renewal capacity. So they use a CD34 positive cells, and using a retrovirus, they transferred the ADA gene into these cells, and this could completely restore the immune system. And earlier patients had to go back to the hospital every 3 months or 6 months to get a fresh infusion of the T cells, whereas these children did not have to go back to the hospital, and they really did very well. And there were 10 children who were cured. But then, unfortunately, later on, when about 20 children had been, who had received this treatment, they found that about 5 of them subsequently had T cell leukemia. And so they had to really stop and look at whether this therapy is really safe or not. But then these children would not have survived at all beyond 2 years. So at least with this therapy, they were surviving. So the US FDA permitted them to carry on the US FDA as well as the European equivalent of FDA allowed them to carry on with this study after looking at the the reason why this could have happened and how they could prevent this. And it was seen that this happened because there was insertional mutagenesis. So retroviruses have the capacity to integrate into the host genome, and they had activated a growth factor like gene, because of which these gamma delta T cells expanded, and this brought about a disease like T cell leukemia. So there were efforts made to prevent this, and they did carry on with the therapy in other children. So this is an example of a young girl who received this therapy, where her CD34 cells were transfected or infected with retroviruses carrying the ADA gene, and she is now leading a normal life, which she would not have been able to do if she had not gone for this therapy. So then, as I said, adenoviruses are being used to a great extent, and there are many examples. I cannot go into the details. There's a shortage of time, but there was one bad thing which happened because of the adenoviruses, where this young boy who was 18 years old, and he had a nitrogen metabolism disorder, and there was a mutation in the gene called ornithine transcarbamylase, and this gene was being repaired with the help of adenoviruses carrying the wild type gene, and he offered himself for this therapy, and he was given a very high dose of the adenovirus carrying the wild type OT gene, and unfortunately he succumbed to this to liver failure because of too much of virus load in this patient. So this was an unfortunate study, but so these are things which did happen, and from which we learned what are the precautions that need to be taken for gene therapy. Now, coming to cancer, since I'm from the Cancer Institute, I'll be talking a little bit more about cancer. And as we know that there are maximum number of trials going on for cancer. So in cancer, there are just three major themes in cancer gene therapy. One is the strategy to kill the tumor cells or to slow down the growth of these cancer cells. So, and we need to develop vectors which are safe, efficient, and targeted. So targeted is the key word. It's really difficult to target each and every tumor cell. So there are efforts being made to target these genes efficiently to the cancer cells. And then finally, the most difficult part for academicians, so people who are working in the lab, is translation of preclinical studies into clinical protocols to test the safety and efficacy and take it ahead in clinical trials. So, as I said earlier, retroviral vectors were being used to a great extent, and the mononeurine leukemia virus was being used mainly because it was very well characterized vector. And the gene vector system which was most widely used for cancer was the suicide gene therapy using the herpes simplex virus thymidine kinase gene with the help of replication incompetent retroviruses. And so let us see what is this suicide gene therapy. In suicide gene therapy, what is done is a gene coding for an enzyme which can convert a prodrug into an active toxic drug is used. So the gene is with the help of a vector, it's transferred into the tumor cells, and then a prodrug is given. So the tumor cell will now start making the enzyme which will convert this prodrug into an active form, a toxin, and kill the cells. Now the added attraction over here is that it not only kills the cell which is expressing this enzyme, but it also kills the neighboring cells, which is called as the bystander effect. So this way you can amplify the effect of cell kill by targeting just a few cells in the tumor. So let us see which are the two suicide genes which are commonly used. One is the herpes simplex thymidine kinase. This converts ganciclovir, which is a non-toxic drug, into a toxic form which is phosphorylated. And there is another enzyme, cytosine deaminase, which is from bacteria. This can convert 5-fluorocytosine, which is non-toxic form, to a toxic form, which is 5-fluorouracil. So both these genes have been used in all these prodrug activation strategies. So in our lab, we decided to use this strategy of using herpes simplex thymidine kinase, and at that time we were using retroviruses. So first we had to establish and characterize a xenograft nude mouse model, because we needed to have a model where human tissue from Indian patient could be used. So we set up a cell line from a human patient, from a patient's tumor, from an Indian patient's tumor, and this cell line grew as a as a cell line as well as a xenograft in nude mice. Then we also made this retroviral vector carrying the HSV-TK gene, and we had a cell line which was making this virus, and we used this cell line for our studies, where the cell line which was making the virus was injected into the tumor in our mouse model, and then subsequently ganciclovir was given. So these were some of the studies. I'll just very quickly go over that. This is a cell line that we made, which we called as NT80 cell line, and this was from a patient with squamous cell carcinoma of pyriform foci. This cell line is being used extensively for many studies. Many people all over the world are using this cell line now. And this, as I said, it grows as a tumor in nude mice. And this is just a long story short, where the tumor cells which received the HSV-TK producing cell lines and followed by ganciclovir showed a great reduction in size compared to the control, which received cells which were not making the virus, and both were treated with ganciclovir. And this is just the pathology. The pathologists will appreciate that these this shows that there is necrosis of the tumor. And these are some of the experiments that we carried out. We used a combination therapy because HSV-TK ganciclovir alone was not able to bring down the tumor completely. So we decided to give IL-2 along with the HSV-TK, and we did some studies, and it showed that there was a better regression of the tumor when we were using a combination therapy. So by the time we were carrying out these studies with retroviruses, there were reports from the, there were reports that a very large study had been done using 248 patients for malignant glioma, and here most of the studies had failed. So although in the mouse model or in vitro studies, we were able to show that it is very effective, and so also this group had shown. But then when they did a clinical trial, they found that the transduction efficiency of the retrovirus in human tumors was very, very low, and so the efficiency was very poor. So this failure of the retrovirus gene therapy study may be due to the low tumor transduction rate observed in vivo in human subjects compared to what we saw in the mouse model. So based on these, retroviral vectors were largely abundant, and we also then switched to adenoviruses, where we made an adenoviral vector carrying the HSV-TK gene, and it also had green fluorescence protein as a marker. So this just shows our cells were transfected with this virus, and it did make the GFP, and it did make the HSV-TK enzyme as well. Now we use this again in our new mouse model. So this is the beginning, where this is group 1, and these are group 2 animals at the start of the tumor. So we start injecting these cells, the virus into the tumor once they are of the same size. And then in the control, the tumor is allowed to grow, and you see the tumor has grown. And here there are different doses of the adenovirus which we used, and we find that the tumor did regress. So this was the adenovirus carrying HSV-TK followed by ganciclovir. So again, to cut a very long story short, we found that the tumor was not really completely regressing, and we wanted to increase the bystander effect and the expression of the transgene as well in these mouse models. So we used HDAC inhibitors, and we found that when HDAC inhibitors were used, the infectivity of the virus had increased. So this is a control where only adenovirus HSV-TK was used. Here the cells were treated first with valproic acid, which is a HDAC inhibitor, followed by HSV-TK. And we found that not only was there more infectivity compared to the controls, but also the expression of the gene had gone up. So somehow valproic acid was somehow increasing the infectivity of the retrovirus, adenovirus into the cells, and also the expression of the transgene was increased. So these are again animal experiments which showed that the tumor did regress completely if the animals were given valproic acid prior to the adenovirus carrying HSV-TK and ganciclovir. So based on these studies, we wrote down a clinical trial protocol where we proposed to use adenovirus carrying the herpes simplex thymidine kinase gene and ganciclovir along with valproic acid for the treatment of oral cancers. This took a lot of time, really. I must tell you how it was really very difficult to write this protocol and get it approved from the ethics committee. But the biggest problem was getting clinical grade vector. So we could not go on with the clinical trials because we did not have clinical grade vector, and we had a vector which had GFP in it, green fluorescence protein, which is not permitted to be used in patients. So this is where the story ended, unfortunately. Now I'll just switch gears a little bit and talk about the other gene therapy trials which are going on all over the world and the success story in gene therapy. Now this is just what James Wilson, who's a very famous gene therapy person, he's editor of Human Gene Therapy, what he said was, the emergence of gene therapy as a scientific discipline in the 1990s stimulated a lot of public hype, a spike in investor speculation, and ultimately a number of clinical trials which were largely unsuccessful. But we learned a lot from these trials, really. So in retrospect, we learned that these first generation technologies were inadequate and the clinical targets were not well thought out. And it became clear that technical challenges of successful cell and gene therapy were much more daunting than one had imagined in the beginning. So preclinical success did not translate to clinical success. What we saw in the animal models or in vitro systems did not really work the same way in human subjects. So the bottom line was that the enthusiasm and hype exceeded what science could support. But there have been quite a few promising results coming out of gene therapy now. The first one is the world's first gene therapy drug to obtain a drug license. So I must tell you that right now we've been talking about clinical trials. So they have to go through phase 1, phase 2, phase 3, and then finally get approval for commercialization. So this was one of the first drugs which got a license for commercialization. That was in China. It is called Gendicin, and this is an adenoviral vector carrying the P53 gene. P53 is a tumor suppressor gene which is mutated in most of the human tumors. So here P53, adenovirus carrying P53 is given to the patients and followed by radiation therapy. So actually this work was going on in the US, but somehow they never got FDA approval, whereas in China they managed to get the FDA approval. The other drug which was approved again in China is Oncorin. So this is oncolytic adenovirus. That means that adenovirus can enter into the cells, multiply and lyse the cells. And this virus, this oncolytic adenovirus can multiply or infect only cells or multiply only in cells which have a P53 mutation. So as I said, most of the human tumors have a P53 mutation or inactivation of the P53 gene. And so it was, they found that this will lyse only the tumor cells. So this is another drug which has been approved by the Chinese regulators. Now the first gene therapy, gene augmentation therapy to be approved by the European Commission was in 2012, and this is Glybera. This was using an adenovirus associated viral delivery for the gene coding for LPL, which is lipoprotein lipase. And because of deficiency in this LPL gene, there are elevated levels of serum triglycerides causing life-threatening pancreatitis. So this was approved in patients in which there was a mutation in the LPL gene. However, this unfortunately had to be withdrawn by this company Unicure, which was marketing this, because they were asked to follow up these patients who had received this therapy for a very long time, and the maintenance cost of doing this follow-up was very, very high. Also, the cost of the drugs was very high. It was US dollars 1 million per patient. So there are very few patients who are really coming ahead for this therapy. So unfortunately, this has been withdrawn, but I don't know whether some other company is going to take it up, and they may not have to pay the maintenance, or they may have to pay the maintenance cost, but they will not have paid for the initial investment for discovering this drug. Another success story is a drug which has been approved by US FDA as well as the European community, and this is called Imligec. Imligec, this has received approval for cutaneous and subcutaneous melanoma in October 15 very recently, and this is again an oncolytic virotherapy where herpes simplex, herpes virus based vector is used. So this herpes simplex virus grows in these tumor cells, and it also carries the GM-CSF gene, which stimulates the immune cells to proliferate. So this has an added effect. So there is a combination of cell death as well as stimulation of the immune system. So Imlygic is probably the first oncolytic vector to receive approval in the US and Europe. So as I said, it's a herpes simplex virus vector which can lyse the cells, and the immune response against these infected tumor cells is enhanced because of GM-CSF, which is also added along with the in this virus. So the third approved gene therapy in Europe is the retrovirus mediated ex vivo gene therapy for patients autologous CD34 positive cells. This is again what we saw earlier, that little girl who was cured because of because the retroviral ADA gene was transferred into the CD34 cells. So this has finally been approved by the European Committee for severe combined immunodeficiency, and this drug is called as Stremvelis, and this works using a virus to insert copies of the ADA gene into the stem cells, that is the CD34 positive cells, and this is engrafted back into the bone marrow where they produce the hematopoietic, they produce large amounts of good ADA, and thereby the T cell function is restored. So there are other promising gene therapy clinical trials which are going on. I talked about the gene therapy products which have been approved, and now we look at some of the upcoming promising gene therapy trials. So one is the non-lytic replication competent retrovirus. So this virus can infect all the dividing cells, which is the tumor cells in the brain, but it does not lyse the cells. So this vector is by the company, it's called TOCA 511, and let's see how it works. So this is injected, if you put it into the tumor, only the dividing cells will get infected because the retrovirus is incapable of infecting non-dividing cells. So it will get infected, and then you allow some time for the virus to grow in this tumor, and then this is followed by, so this also carries a cytosine deaminase gene. As I told you, that's a bacterial prodrug activation gene, which converts 5-fluorocytosine to 5-fluorouracil, and then it kills the cells which are expressing it. So this was, this is given to the patients after surgery. So after the brain the tumor is resected in the cavity, these cells, this TOCA 511, which is the virus, is injected, and then it is allowed to grow in the brain for some time, and after that this is followed by oral TOCA 5FC, which is 5-fluorocytosine, and so here again, in addition to killing the cells, the killed cells stimulate the immune system. So there is an added this thing where the immune system is selectively activated by the local tumor killing, with resultant systemic anti-tumor immunity. And from a trial which is going on, they found that the median survival after recurrence of such patients after surgery is 7 to 9 months. Whereas after this therapy, they find that the median overall survival is 13.6 months. So it's increased slightly, of course, but there is an increase. And the probability of survival at 24 months is 31.6%. So that means at least 31% of the patients survive for 24 months compared to just 7 to 9 months. And in a subset of these patients, they found that the overall survival was 14.4 months instead of 7 to 9 months, and the probability of survival at 24 months was 40%. So 40% of the patients could survive at the end of 2 years. So this is another promising therapy, but it has still not been approved. So it's still waiting approval, and a large number of studies, trials are going on for this. So basically it was also important that it had favorable safety profile and was well tolerated by the patients. Now another very promising therapy which is coming up is the CAR T cell therapy, which is chimeric antigen receptor T cells. And this was designated as a breakthrough therapy by the US FDA in 2014. And there are a lot of trials going on. This looks very promising. So initially it was done for acute lymphoblastic leukemia patients where the T cells were removed and genetically manipulated, and then they were given back to the patient. So the receptor was modified in such a way that it recognized the CD19 protein on the B cells, and it killed the B cells. And here the results look very promising again. So this is one of the diseases which is very difficult to treat, and so this looked very promising. I'll just tell you, I think, the next slide. Okay, so here what is done is the T cells are collected from the patient's own blood, and then they're genetically engineered to produce special receptors which are called the chimeric antigen receptors, CARs, on the cell surface. And these CARs allow the T cells to recognize specific antigens on the tumor cells. And in this case, it was the CD19, which was the B cells. So these were given back to the patient, and they could kill all the CD19 positive cells and thereby bring down the disease. So the expanded population of CAR T cells is infused into the patients. After infusion, the T cells multiply in the patient's body, and with guidance from their engineered receptor, recognize and kill cancer cells that harbor the antigens. So Carl June is one of the persons who was in whose lab in Philadelphia, UPenn, this study has been going on, and this looks very promising. Now a company has taken over and is doing this. Now this is the first patient. This was a patient who had no hopes of surviving, and she had B-cell leukemia, and she was taken as a special case, and the US FDA permitted them to carry out this trial in this little girl, because the parents were very insistent. They wanted this therapy to be tried for their child. They said that we don't care if something goes wrong, but we want this child to get this therapy. And fortunately, she was cured of leukemia using this therapy in 2015. So now in August 2017, US FDA has approved CAR T cell therapy that genetically modifies a patient's own cells to help them combat pediatric acute lymphoblastic leukemia, which is the most common childhood tumor. And there are some studies which are initiated now, right now, in India as well, and we hope that this will be a therapy which will not be just for leukemias but also for solid tumors. And so this was the first gene therapy to be approved by the US FDA. And then there are other examples where hemophilia is also showing some promise, where the factor 9 gene is either injected directly into the patient's liver or through mesenchymal stem cells, which are first infected with the virus. Here the adeno-associated virus is used, and then these mesenchymal stem cells are given to transferred back to the patient. So this also has shown some promise, and there are clinical trials going on for this. So I think two techniques which have given a boost to cell and gene therapy today is Yamanaka's discovery of inducible pluripotent stem cells, where you can take the patient's fibroblast skin cells, grow the fibroblast, and make inducible pluripotent stem cells there. And the other one is development of gene editing techniques using CRISPR-Cas9 system. So there's one very, I'll just give one example. Oops. Oh my God, what happened? I'm sorry, if I can go. Sorry. Can you, the slides, I think. So there's just one example of gene editing that I want to give. Go to the, no, one before. Yeah, this is the one. Thank you. So this is a very interesting story. There was this person called named Timothy Brown. He is now known as a Berlin patient. He was suffering from AML, acute myeloid leukemia, as well as HIV. And he was given a bone marrow transplant for his AML. And just by chance, he got a bone marrow transplant from an individual who was homozygous for naturally occurring 32 base pair deletion in CCR5, which is a receptor for HIV. This is a co-receptor for HIV. And then following this success of transplantation, bone marrow transplant, they've discovered that he had no HIV at all. He had no, his AIDS was also cured. So this is because he had bone marrow cells from this patient, from this donor who had absence of this receptor, which was used as a HIV co-receptor. So now they are trying to do gene editing. There are studies, clinical trials going on where they're trying to modify the CCR5 gene, and that might be the cure for cancer, HIV patients. So finally, I think I'm convinced that gene therapy is emerging as a promising way to treat a number of diseases for which there was no treatment available. And success continues to be achieved in experimental models using AAV, adeno-associated virus, and lentivirus. So these, out of all the viruses that were used, these two seem to be very promising, and a lot of studies are now going on with AAV and lentiviral vectors. Engineered blood stem cells using lentiviral vectors has shown promise in human trials. And a large number of pharma industries are now getting interested and investing in, once again, they're investing in gene therapy products, which is a good sign. So we have learned from the many human clinical trials what we would never have learned from animal studies. So what happens in animals is not really, but then we have to have some model where we can use, I mean, to show initial listing that it works. So it has to be done, but it does not mean that if it works in animals, it will work in the humans. And now it is time to harness the power of gene therapy to treat cancer and some of the rare diseases. Thank you. So if there are any questions.
Thank you so much, madam, for sharing the pioneering work on gene therapy. I mean, I was part of the lab at that time. I remember the excitement when there was a decision of change of the vectors based on the clinical trial, and also the IL-2 therapy and the promising results that it gave. These are all colleagues who are now working in various pharmaceutical companies in the US for madam students, and have really harnessed this potential of the technology that they learned. I think you all will agree that as madam has rightly pointed, whatever is conducted so successfully in the animal models may not be what is seen in the human studies. But what actually for me, as I was learning throughout this experience, though my project was miles away from what gene therapy was conducted, and the facilities that were built at ACTREC with so much of precision, that despite of all that, and with the wonderful project which was done, which was formulated with Professor Anil De Cruz, was that a clinical grade vector was something that was really falling short of. So if there are any queries or any clarifications or any suggestions that the audience may have, for madam, where you would want to have a further conversation. Yes, Dr. Puneet.
What are the type of molecules, and how do you actually decide what is to go site specific and what is?
Specific is mostly in cancer. In tumors, if you can inject directly into the tumor. The systemic would be mostly for blood cancers and for other solid tumors where you can't access the tumors.
Site specific is being promoted, and the uptake of the molecule.
Better because it doesn't spread everywhere. So, but that can be done only in site specific. Even for hemophilias, they're injecting it into the liver so that the liver starts making the factor 9. But I don't know which one is better. But I think it's case by case. It depends on the disease. It depends on how easily you can access the organ where you want to inject.
That this is a capacity building forum, and you've worked in India and you've really seen it all, and it has been an era. What do you think? What is the message? I mean, in a clinical setup like ours, where there's a lot of, I mean, you talked about CAR therapy and talk and so forth, and you mentioned, emphasized on human trials being one of the aspects, and also the ethics component of it. So what message would you want to give us in terms of the capacity building, not for us but also for all those who are listening to you?
Well, I think we definitely need to develop. I mean, these are not very simple techniques, as you know. These are mostly, of course, molecular biology based, but they need to be done properly. So we need to train people, and I think we should go for some of these, because once these therapies come in the market, once these drugs come in the market, they are going to cost us a lot. So if we develop it in-house, if we develop it in India, I think the cost will definitely come down. And it is possible. It's nothing is impossible.
The challenge is...
Yeah, so especially the CD4, the CAR-T therapy really looks very promising. And I'm sure it's going to come up not just for leukemias, but for solid tumors as well. So I would think that we need to do a lot of basic work, of course, before we think of doing clinical trials. So first we do basic works, we have animal models. We can't do away with animal models. We have to have animal models. And then go on to planning clinical trials. And we need to collaborate with the clinicians a lot to understand the clinical aspects. In this country, unfortunately, the scientists don't have too much of a dialogue with the clinicians, which is bad, I think. So we should so even in Tata Hospital, I think I had a very tough time, convincing the doctors that we need to look into this. We need to really plan a clinical trial for gene therapy. So it was difficult to make them understand. In fact, there was Dr. Sultan Pradhan, who's a very famous surgeon. He's much senior to me. When he was part of this study, he said, first you come and teach me what is DNA, what is RNA, how do I go about it? So things are not the same now. I think most of the doctors now know what is DNA, RNA, PCR and all that. But the earlier generation didn't know, so it was difficult. But one needs to talk to the clinicians before planning anything that you hope to